For today's webinar. My name is Jim Sergi, and I am the president of CSSI Life Sciences, a fully integrated global life sciences company whose mission is to accelerate research discoveries to a commercial success. CSSI Life Sciences possesses deep subject matter expertise and a proven track record with drugs and biologics, including vaccines. Today's webinar, Innovation in Infectious Disease Research, Challenges and Opportunities, will be led by Dr. Heidi Nelson-Kayerle, Executive Director of Preclinical Services at CSSI Life Sciences, and Dr. Raymond Goodrich, Executive Director of Infectious Disease Research Center, as well as Professor in the Department of Microbiology, Immunology, and Pathology at Colorado State University. Both Dr. Nelson-Kayerle and Dr. Goodrich biographical summaries are included. Prior to starting today's webinar, I would like to do a little housekeeping. The goal of this webinar is to discuss topics of interest in the field of infectious disease research and related emerging threats to public health and safety. In addition, we hope to foster dialogue on various ways that we can convert the challenges that we collectively face into opportunities to improve patient care and patient outcomes. The objective for today's webinar are shown in this slide. Today's presentation is being audio recorded and will be archived. You will receive a copy of the recording and will be able to access the slides following today's program. You will also have the opportunity to submit text questions to today's presenters by typing your questions into the question box, which is located on the top right-hand corner of your screen. You may also send your questions at any time during the presentation, and we will collect these questions and address them during the question and answer session at the end of today's presentation by Dr. Goodrich. Lastly, if you have a free minute at the end of the webinar, please consider registering for Biomark's upcoming IDR Talks Symposium. A link will be provided for you to register at the conclusion of the webinar. I would now like to begin our program. Dr. Nelson-Kayerle will begin the program, followed by Dr. Goodrich. Dr. Nelson will discuss development of vaccines for infectious disease. Please welcome Dr. Nelson-Kayerle.
Thanks, Jim. I wanted to start my presentation with the FDA definition of a vaccine. According to the FDA, a vaccine is an immunogen intended to stimulate the immune system to result in the prevention, amelioration, or therapy of any disease or infection. Another important term in vaccine development is adjuvant. An adjuvant is an agent that is added to or used in conjunction with a vaccine antigen to augment or potentiate and possibly target the specific immune response to an antigen. Again, according to the FDA, a vaccine may be composed of a variety of different materials. Live attenuated preparation of bacteria, viruses, or parasites, inactivated whole organisms, living irradiated cells, crude fractions or purified immunogens, including those derived from recombinant DNA in a host cell, polynucleotides such as plasmid DNA vaccines, living vectored cells expressing specific heterologous immunogens, and therapeutic cancer vaccines, which induce immune responses to tumor-specific antigens. Next slide. This shows a cartoon of the vaccine development pathway. Starting on the left, it begins with an unmet medical need as well as technical feasibility. That is, a target antigen has been identified and the protection mechanism is understood. During the pre-clinical development phase, The antigen is further defined and purified. The vaccine formulation is developed. Immunology in relevant animal models is studied. Experiments on mechanism of action are conducted, and toxicology evaluations in animal species are performed. And in addition, production of GMP lots for the phase 1 clinical trials are also performed. During clinical development, phase 1 trials are conducted for safety in small numbers of participants. Phase 2 trials include more participants and look for safety as well as efficacy. The phase 3 vaccine trials tend to be quite large with thousands of participants, and again look for both safety and efficacy. These trials will assess correlates of protection where possible to see if the vaccine is actually protecting the population from the pathogen. During the clinical trials, as those are being performed, the GMP processes for commercial manufacturing of the vaccine product are also completed. During the post-licensure phase, the cost and effectiveness are assessed for reimbursement. An immunization program is implemented, and pharmacovigilance or continuous monitoring of adverse events is conducted. And frequently for clinical trials, frequently for vaccines, phase 4 studies are also conducted to look for efficacy in larger populations. Next slide. FDA CBER, or Center for Biologics Evaluation and Research, is the department that regulates vaccines. Next slide. And the regulatory path starts with the Investigational New Drug Application, or IND. This document describes the vaccine. It includes descriptions of the manufacturing processes and the quality controls put in place for testing the release of the GMP material. The IND also includes the animal safety data, including immunogenicity, and the proposed human clinical protocol. The Biologics License Application, or BLA, is submitted to FDA after phase 3 clinical trial completion. And this application includes the manufacturing data as well as the safety data from the preclinical studies and the clinical trials. And the BLA also includes the labeling. Finally, there is a vaccine committee, the Vaccines and Related Biological Products Advisory Committee. This is a group of non-FDA experts, including scientists, physicians, biostatisticians, and consumers. And this committee may advise FDA regarding the safety and efficacy of the vaccine for the proposed indication once FDA has reviewed the BLA. Next slide. So during the IND vaccine development, the animal studies, several of them will need to be done under good laboratory practices, or GLP. This is basically a quality system for execution of the non-clinical safety studies and ensures data integrity for FDA so that they're able to make informed risk and safety assessments. The definitive preclinical studies must be GLP compliant, and the GLPs cover personnel, facilities, documents, test and control articles. Next slide. Similarly, on the manufacturing side, for the IND, good manufacturing practices, or GMP, is important for quality assurance for consistent product manufacturing. And the GMPs cover controls for with a quality standard appropriate for the developmental stage of the vaccine. These controls cover the identity, safety, purity, efficacy, potency, stability, and consistency of the vaccine product. Next slide. So for the preclinical phase, discovery looks at identification and validation of the antigen, in vitro efficacy studies, biological screening assays may be included in discovery, as well as animal efficacy models looking at reactogenicity and immunogenicity. Next slide. For the vaccine animal studies, the species selection is an important decision. So some of the criteria to think about are listed here. The species should elicit a similar immune response as what is seen or expected in humans. The adjuvant, if there is one, should demonstrate a similar immunological effect as in humans. And the animal model should reflect the course of infection in humans. And some things to consider when looking at your species selection is the feasibility of the route of administration, the availability of naive animals for the pathogen in question, as well as the availability of reagents for immunological analyses. Next slide. For the IND during vaccine development, a number of animal safety studies may be conducted, and these are listed here. So there's an immunogenicity study, and if there's an animal model for the pathogen, that study can also incorporate protection. A single-dose toxicity study, which may also be folded into the repeat dose tox study. Local tolerance, which also can be folded into the repeat dose study. Safety pharmacology studies, again, could be standalone or part of the repeat dose. And the safety pharm studies look at cardiovascular, CNS, and respiratory systems. If you're developing a live attenuated or DNA vaccine or using a new adjuvant, biodistribution studies may be important, and reproductive tox studies, depending on the target population, may also be needed. Next slide. So the immunogenicity protection study evaluates the animal immune response to a vaccine relevant to protection. These studies can be conducted not under GLPs, so non-GLP, and during the conduct of the study, the production of antibodies should be evaluated. That could be IgG, IgA, neutralizing antibodies, or bactericidal. Also to be studied is cell-mediated immunity, seroconversion rate, and immune memory. And again, if the animal model exists for protection, that should be looked at as well. Next slide. The single-dose toxicity study is a GLP study that provides preliminary tolerability and safety data. It's typically performed in rodents, such as mice or rats, and it's an acute or single-dose toxicity study using the full human dose. Oftentimes, this single-dose study is incorporated into the repeat dose study. However, if there's significant intrinsic toxicity or if there's an immune response to the first dose that alters reaction to the next dose, for example, generation of neutralizing antibodies, in either of those situations, a standalone toxicity single-dose study will be needed. Next slide. The repeat dose toxicity study is your pivotal study. This will be done under GLP, and it helps establish a safe human dose and looks at vaccine safety. Again, the full human dose is typically administered. the number of doses administered should exceed what is expected in the clinical trial using the N+1 rule. In the animal studies, the dose intervals may be compressed compared to the human trials, for example, 2 to 3 weeks, depending on the kinetics of the immune response in the animal. These studies will look at serology, that is, the antibodies, T cells, cytokines, other correlates of protection. Full histopathology will also be conducted, and the study will include a recovery group to look at persistence and immune response toxicities. Next slide. Local tolerance is another GLP study. This one evaluates tissue reactions at the site of administration using histopathology. It's often included into the repeat dose study, but if necessary, a standalone study can be conducted. These are typically done in rabbits, and that gives you more information on the kinetics and resolution of the reaction. Adjuvants typically or often produce local reactions, and so if you're using a novel adjuvant, a local tolerance standalone study may be required. Next slide. So for the IND submission, there's a section on chemistry, manufacturing, and controls, or CMC. Next slide. And this shows a cartoon of a sort of typical vaccine manufacturing process. There's upstream processing, which includes strain selection and growth, as well as downstream processing, which includes purification, inactivation if it's required, formulation, and fill finish. Next slide. If you're working on a viral vaccine, here's some information on the upstream processes. There are 3 stocks that are involved in the viral vaccine manufacturing. The master cell bank, which is typically a mammalian cell bank. A master virus seed stock, which is generated using the master cell bank. And a working virus seed stock, which is generated by passaging the master virus seed stock. In other words, expanding that for vaccine production. All 3 of these stocks need to have full GMP characterization, looking for purity, identity, potency, specificity, biosafety, including attenuation, tissue tropism, and shedding, sterility, looking at both adventitious agents as well as bacteria and fungi, replication, competency, genetic stability, and long-term stability. Also, you have to document passage history and derivation of these stocks. Next slide. Downstream, you've got your post-filtered harvest or final bulk, as well as your final filled product. These also need to have GMP characterization, looking at sterility again, safety, identity, purity, and potency. And this image on the right is the Biomark facility in Fort Collins, Colorado, part of Colorado State University. It's a high containment GMP manufacturing facility that can handle BSL-2 and 3, select agents, and spore formers. Next slide. After your IND has been submitted, the vaccine moves into the clinical development phase. Starts with a phase 1 clinical trial with approximately 20 to 80 participants typically, and that looks at the safety of the vaccine. The phase 2 trial follows phase 1, and you have hundreds of participants, and that study looks at efficacy of the vaccine. Phase 3 trials typically have thousands of participants and look at the safety, efficacy, and dosing of the vaccine in a larger population. On completion of the phase 3 trial, the BLA is submitted to FDA, and frequently after FDA approval, there's a phase 4 study conducted for vaccines, and that's looking at the long-term effectiveness of the vaccine as you start to administer this to a much larger population. Next slide. These are my contact details. Feel free to reach out to me should you have any questions. And this concludes my presentation. Back to you, Jim.
Thank you very much, Heidi, for that presentation. I would now like to turn the webinar presentation over to Dr. Goodrich, who will present an introduction to infectious disease research at Colorado State University, the Infectious Disease Research Center at CSU. Dr. Goodrich?
Yes, thank you, Jim. What I would like to do today, as Jim indicated, is give you an overview, a high-level overview of the Infectious Disease Research Center at Colorado State University. In particular, we'll be talking a lot about BioMark, which is the Biopharmaceutical Manufacturing and Academic Resource Center, which is part of the Infectious Disease Research Center. As this first slide indicates, our mission is really to provide, to maintain, to support, and enhance the ability of different research groups to conduct research that involves high containment biological organisms. We do that based on support that comes from a foundational standpoint, faculty-based research programs. There's a very long history of infectious disease research that has been ongoing at Colorado State University throughout its history. On that foundation are built three main pillars of activity: the BioMark, Biopharmaceutical Manufacturing and Academic Resource Center, which operates as a cGMP facility for manufacturing of diagnostics, therapeutics, and vaccine candidates; the Regional Biocontainment Lab, which operates as part of the National Institutes of Allergy and Infectious Diseases network for biocontainment facilities in the United States; and the Research Innovation Center Incubator, which houses private industry, early phase and mid-phase companies that are developing technologies in this space. I'm going to tell you a little bit about each of those as we go through the presentation. We have the next slide, please. So the Regional Biocontainment Lab is the part of the facility that houses our BSL-3 level facilities. These are facilities that are specifically designed from the standpoint of filtration, air handling, containment design issues to allow for the handling of agents that protect both the researchers as well as the prevent exposure of these agents to broader populations while this research is ongoing. These are housed within a facility, was constructed in part with support through the NIAID several years ago, back into the 2005 timeframe here at Colorado State University. The next slide. This gives you an idea of the network of facilities that are capable of handling both BSL-3 and BSL-4 agents. There are two facilities in the U.S. in Boston and in Galveston who are part of this network capable of handling BSL-4 level agents. Colorado State University, as indicated here, is one of the BSL-3 facilities in the network, and as you can see from the map, we are the organization that's located furthest west in the United States with these capabilities in place. The next slide. So the Regional Biocontainment Laboratory really contains the biocontainment facilities for research on biodefense and emerging infectious disease agents. It houses the animal models core, which includes multiple animal handling rooms that comprise that core. We handle animals such as rodents, mosquitoes, large and small animals ranging from goats and sheep to camels, rabbits, and ferrets. The facility is designated at a super complex level by the CDC community in their ranking of security complexity. What that means is that we have the capability of handling these types of agents in a research environment with the suitable facilities, the suitable training for our personnel, and the suitable infrastructure and procedures for the handling of these agents in an appropriate format. Next slide. The BSL-3 space within the IDRC comprises approximately 50,000 square feet of facility. It houses the research on CDC select agents and Tier 1 agents, a list of some of the other agents that our research faculty and collaborating partners are actively involved in is described in this slide as well. I won't bother to go through each one of these individually, but I think you can see a wide variety of agents ranging from tuberculosis to bacterial agents to viral agents that represent both existing as well as emerging viruses that we handle routinely in the facility. The next slide. Biomark is an FDA-inspected, not-for-profit service entity at Colorado State University that specializes in the production of biological products that require this high containment facility capability. This includes the Biosafety Level 3 and CDC Tier 1 select agents, as well as spore-forming microorganisms. Biomark really offers product life cycle support for manufacturing in biological therapeutics, vaccines, and diagnostic products. The next slide. This gives you an overview of some of the services that are provided, and those services are available for a variety of clients, whether they're academic research laboratories that have this need, government laboratories that have this need, or virtual startup, mid-level, or large pharma companies that have this need in the private sector. The facilities basically exist to help support technology transfer, project management, in-house testing, process and method development, which takes the product from concept to creation. It's one of the few high containment biological manufacturing CMOs in the US capable of BSL-2 and BSL-3 cGMP biological manufacturing. It is FDA and CDC inspected and audited for EU compliance. In addition to these capabilities, under the Infectious Disease Research Center, the Biomark operation is also able to support preclinical and early phase 1 clinical study work. A large part of that is done in collaboration with the strategic partnership was announced earlier this year with CSSI Life Sciences. Given their history and capabilities in this area, the partnership made sense to allow us to advance products which are in the manufacturing or pilot manufacturing mode in the Biomark facility into the early phase 1 clinical trial space. Next slide. The Research Innovation Center Incubator, which is also located in our facilities here at Colorado State University, really has the mission of growing the bioscience industry in northwestern Colorado. And we do that by providing state-of-the-art lab space and capabilities that allow companies to leverage their capital and research dollars without having to build these infrastructures on their own, utilizing capabilities that exist internally. It also provides an environment because of the commingling of faculty research, students who are trained in these fields, for companies to resource their capability needs with regard to employees, and also to research their capability needs with regard to sponsored research agreements or information consultation that's required to help advance their product development portfolios. There are several evaluation criteria in an application that tenants have to fill out prior to entering into the system at CSU, and if there is a good fit, after review by an internal committee, they are invited to take residency in the Research Innovation Center facility, which is located in the Foothills campus at Colorado State University. Next slide. The goal of this facility, as I mentioned, is really to provide life science technology companies with an opportunity to collaborate with outstanding university faculty and to establish a presence on campus. It really works in the early phases of the development of new programs by providing access to office space next to flexible wet lab space, access to laboratory animal services and a state-of-the-art vivarium, access to CSU faculty, students who are trained in this field, and a shared infrastructure that allows groups to access capabilities within the CSU library system or major equipment cores. The relationship that we have with groups such as Innosphere, which is run by the City of Fort Collins as a technology incubator, and CSU Ventures, which is our IP and licensing arm of the Colorado State University, allows us to establish those relationships with companies that are interested in having those support and advisory services to help them in the early stages of development of their company business plans and commercialization of their products. The next slide. What this really shows you, I think, is the fact that we are active in the continuum of response when it comes to existing and emerging diseases, whether that is identifying early warning, pre-event opportunities to identify new agents as they emerge, involved in the early detection of those events, and then also being involved in the rapid response in the development of vaccine and therapeutic countermeasures in order to address emerging public needs. And this very much fits not only with the mission of Colorado State University as a public institution, which is focused on research, education, and outreach into the community to address needs in the public health sector in the United States, but also fits with the original intent with which these investments were made to develop these capabilities to allow for sites in the United States for the handling of these agents, research and education related to them, so that we might be able to address broad public needs in the infectious disease space that result from the entry of these agents into human populations. In the next slide, I just summarized a few of the types of programs funded through groups like the NIH and the DoD that describe just a small portion of the faculty-based research programs that are ongoing. These are projects which are active and ongoing within the arthropod-borne and infectious disease laboratory, which is located again in the Foothills complex of the Colorado State University campus. It gives you some idea of the diverse types of programs that are ongoing within the community of faculty-based research that's located here at CSU. In the next slide, as was mentioned earlier, I would welcome you to please consider registering for our upcoming event that will be held June 7th to 9th at Colorado State University, Innovation in Infectious Disease Research, Challenges and Opportunities. This represents, you'll be able to see from the website that's indicated here, idrtalks.colostate.edu, a wide variety of speakers and participants from around the country who are involved in academic research, involved in government laboratory research, as well as involved in private industry research, ranging from the very small startup companies that are part of our RIC incubator to very large organizations like Merck who are developing technologies in these areas. There are a variety of parts of this program that are described in the agenda, as well as some background on the speakers that we currently have lined up for this. We welcome you to attend this conference and would welcome your participation. With that, I'm going to turn it back over to Jim.
Thank you, Dr. Goodrich, and thank you, Heidi. What we would now like to do is to begin the question and answer session. As a reminder, you have the opportunity to submit your questions via text to today's presenters by typing your questions into the question box, which is located on the top right-hand corner of your screen. What I'd now like to do would be to begin with the first question. The first question is directed to Dr. Goodrich. And the first question really is, it's coming from Research Triangle Park in North Carolina. And that question is, what is the difference between a BSL level 2 and a BSL level 3 agent? Dr. Goodrich?
It's a good question. This is really a system that was established several years ago to classify organisms in terms of their capability to cause infection and the outcome that those infections cause in human populations, whether they're transmitted in an aerosol form or in a contact form. So they're classified according to their capability to be transmitted by different routes, as well as by the consequences of those infections. The higher the level, the more stringent the containment requirements have to be for handling those agents. I describe it to people as sort of being the equivalent of a Richter scale. As you go from a BSL-1 to a BSL-2 to a BSL-3, it's not simply a matter of a difference of one in terms of the requirements that you have to put in place to contain these agents. It really is 100 to 1,000-fold higher in terms of air handling requirements, personnel training, safety and security measures, SOPs or procedures that have to be put in place. And those are, as I indicated, some of the capabilities that we've built here at Colorado State University as it comes to specifically the handling of BSL-3 level agents that are required for the development of therapeutic, diagnostic, and vaccine approaches to dealing with these types of diseases.
Thank you. A follow-up question to that is, what scale of manufacturing capabilities does Biomark have for vaccine development?
Sorry, right now that depends upon the type of agent. We have a bioreactor assembly that can be scalable, I believe, from 20 to 200 liters in size. There are also our staff capabilities and research programs have been involved in some of the continuous manufacturing approaches that would allow you to scale size and availability of vaccines to larger levels if that is required. Generally, we operate, however, in the space that provides initial pilot batches as well as batches that would support studies on the order of a phase 1 clinical study. The unit is not intended, for example, to be a place where you would do production of very large scales of very well characterized agents as a supplement to existing manufacturing. The intent is more to focus on some of the more novel approaches to translate those into production methods that can be scaled up and to demonstrate that scale up in the initial phases of pilot and clinical phase 1 level production volumes that are required.
Thank you, Dr. Goodrich. Another question for Dr. Goodrich is, have you made any vaccines from virus-like particles?
Yes, that is one of the focuses of research that is ongoing. We announced earlier this year publicly a project which is funded through the Joint Vaccine Acquisition Program, and that involves a virus replicon particle technology for the development of a Venezuelan equine encephalitis virus vaccine. And that is a program that we are currently running and producing initial batches of material along the lines of what Dr. Nelson Caerley indicated earlier that are required to move that candidate into the phase 1 clinical trial mode in human subjects. So that is a particular platform and technology capability that exists here within the Biomark operation.
Thank you. We have a question for Dr. Nelson Caerley from a participant in Chicago, and I'll try to combine the questions that were asked into one. And the question is, how many animal studies are required for an IND, and when do animal rules apply, and can you describe the animal rule provision?
Absolutely. So the number of animal studies that are needed for vaccine development for the IND submission Minimally, you're going to need to do an immunogenicity protection study and a repeat dose toxicity study. Again, that repeat dose study has to be done under GLP. Other possible studies, depending on what your vaccine product is, may include the local tolerance study that I discussed, single dose toxicity, safety pharmacology, again, if there's any indications that there may be adverse effects on the cardiovascular, respiratory, or CNS. And then biodistribution and shedding, again, if it's a DNA vaccine or a live virus vaccine. Regarding the animal rule, that pertains to the FDA allowing animal studies to stand in the place of human trials, human efficacy clinical trials. So if the pathogen is severe enough or rare enough that it's impossible to ethically conduct a clinical trial or to do a field trial, if those criteria are met, then FDA allows for the pivotal animal studies to replace the human phase 2 trials.
Thank you. And in other words, one of the questions that also came to us from San Francisco is, are rabbit studies always necessary for a vaccine IND submission, or can other animal models substitute?
So definitely you want to choose an animal model that meets the criteria that we discussed earlier. Similar mode of infection as to a human, and with a similar immunological response as what you'd expect to see in a human. Rabbits are often used for local tolerance studies, and that has to do with a C-reactive protein that's a good marker of immunological response, and rabbit studies are very conducive to using that biomarker. But rabbits can also be used for your pivotal tox study, and again, it gets back to the volume of the dose that you'll be using. If the volume is such that it's impossible to put into a rat, for example, rabbits can certainly be used.
Thank you. A question for Dr. Goodrich is, what makes biomark manufacturing unique and distinct from other similar commercial operations? And is biomark capable of producing, manufacturing viruses if it's only a BSL level 2 agent?
The answer to the first part, what makes biomark manufacturing site unique and distinct, is, I think, several things. First of all, it does operate as a CGMP manufacturing operation for high containment biological manufacturing at the level BSL-3 for agents that require that type of containment. Secondly, it exists within an environment of academic research where there are active programs that are going on in the environment, where you have both a faculty-based population, a student-based population of individuals who are trained in these areas, who are knowledgeable in these areas, who oftentimes are conducting research that's on the cutting edge of understanding not just how to make a vaccine, but how to do that in an efficient and effective way, oftentimes employing new techniques or new methodologies in a translational mode that can be scaled up further into larger scale production. So it's an environment where innovation is really fostered in a lot of ways, because that's part of the fundamental of our mission more broadly for research and education. Can we manufacture agents that require only BSL-2? Absolutely. in many ways because, again, as I mentioned earlier, in the difference between BSL-3 and BSL-2, it's an easier proposition because you don't have as many containment methodologies that are required with the BSL-2 agents as you may have for BSL-3 agents. The fact that we're able to conduct research with BSL-3 level agents, again, several orders of magnitude higher on those requirements, also means that we have space, we have facility that we could set aside specifically to do work with BSL-2 level agents as well.
Thank you. A similar question that was posed is what does it then mean that Biomark is a not-for-profit entity that is associated and affiliated with CSU, Dr. Goodrich?
Well, the facilities were built in part with public funding, in part with federal government funding, and it was built with the intent of being able to serve the public needs. So the shareholders that we answer to are the public in the state of Colorado, in the community of Fort Collins, in the state of Colorado, and more broadly in the United States. You may even expand that more broadly to our global community, where infectious disease issues are a concern to them and to us as well. So our main focus is in undertaking programs and projects that serve that public need. Being not-for-profit does not mean that you're for loss. It simply means that the funds that we generate as a result of our operations are reinvested in our students, in our faculty, in education and research programs that help to advance the public interests as they relate to infectious diseases and being able to develop methodologies that address those in order to improve public health. That's really what it represents.
Thank you. A question for Dr. Nelson Kayerle. When is the USDA involved in vaccine development?
So the USDA is involved in development of vaccines for animals, so agricultural animals or companion animals. Otherwise, all the human vaccines go through FDA.
Thank you. There are no other questions that have come in from our participants today. I would now like to ask Dr. Nelson Kayerle and Goodrich if they have any concluding comments.
I have nothing else, Jim. I think we're good for now. I want to thank you and thank everyone. It was a pleasure being with you today.
Thank you, Dr. Goodrich. I would also like to remind everyone to please consider registering for the IDR Talks Symposium at idrtalks.colostate.edu registration. If you have any other questions or comments, please feel free to follow up directly with Dr. Nelson Kayerle and Dr. Goodrich at their contact information, which is provided on this slide. Lastly, you will receive a follow-up email within 48 hours with a link to view a recording of today's presentation, as well as to access the slides from today's webinar. And on behalf of CSSI Life Sciences, Biomark, and our presenters, Dr. Heidi Nelson Kayerle and Dr. Raymond Goodrich, I'd like to thank you for joining us today. Thank you.